Welcome to another webinar by Technion Australia, where we give you the opportunity to tap into the world of our scientists and researchers and alumni who for over 100 years have been developing applications and research that is shaping the world. In fact, exactly 109 years ago today, we celebrate the laying of the cornerstone of the Technion, which is Israel's first university. This symbolizes the first step of training and educating the future states, engineers, scientists, entrepreneurs, and change makers. My name is Ori Danieli, and I am the CEO of Technion Australia, which is the representative arm of the Technion, Israel's Institute of Technology. With many of Israel's greatest inventions and discoveries credited to the Technion, it is often regarded as the backbone of Israel's claim to fame as the startup nation. In Australia specifically, we're part of a global network with a mission to support the Technion by encouraging philanthropic support and promoting research collaboration. I want to start by acknowledging our event partners today, which is the University of Sydney, which is Australia's first university, established in 1850, and among the world's leading universities. And our second partner today is Meeting for Minds, which is a not-for-profit organization and platform for sharing knowledge and collaboration and understanding of mental illness and disorders of the brain. On that note, we are very honored today to be hosting three leading medical and mental health experts. Professor Ian Hickie from the Sydney University's Brain Institute, Professor Dorit Ben-Shachar, who is from the Department of Neuroscience and Biological Psychiatry at the Technion in Israel, and the discussion will be moderated by Dr. Norman Swann, who is ABC's medical journalist and among Australia's leading medical journalists. They will share insight into a brain with mental disorders. As always, our webinars are meant to be interactive, and you're encouraged to ask questions and click on the Q&A button at the bottom, and our speakers will do their best to answer. On that note, I think it's time to get the show on the road, and it's my great privilege to welcome Dr. Norman Swann, who will moderate the conversation and formally welcome our speakers. Thank you. Over to you, Norman.
So thanks, Ori, and thank you very much to you all for joining us. Thanks, Ian, and thanks, Dorit from Haifa. Dorit, let's start with you. And why did you get interested in these mitochondria, which are really the energy powerhouses of cells? They're involved in aging. There are lots of diseases where they're implicated, including muscle wasting as you get older. Why are you looking at them in the brain?
Our hypothesis was, actually, I was working on other things, and when I joined the psychiatric department, I was working on, pure science, nothing to do with clinicians and clinical aspects, and so I joined the psychiatric disorder, and they were very bored from what I did. It didn't interest them. So I decided to do something clinical. And since I was working with mitochondria, I thought maybe I'll take some mitochondria from patients, psychotic patients, and try whether there is any change in the first complex, any dysfunction in this mitochondria comparing to healthy controls. And my hypothesis was that neurons are heavy consumers of energy and highly dependent on it, and that abnormal energy production in neurons can lead to distorted the neuronal network connectivity and thereby to abnormal behavior. So I took 5 patients, blood from 5 patients with acute psychosis and 5 healthy subjects, and measured some functions of the mitochondria, and I saw that they were completely abnormal and impaired. So I thought maybe it's a beginner luck, and I took 5 more, and then I saw the same thing again. So this led me to this road of mitochondrial research. And actually I think we were among the very first to show that there is this function of mitochondria, this powerhouse brains of in schizophrenia, not only in brain but also in peripheral tissues. Every tissue that we measured was had showed this dysfunction.
So just to clarify, so somebody with schizophrenia, or in this case psychosis, it wasn't just in the nerve cells where you had this powerhouse problem. When you actually looked at any part of their body, they seem to have the same issue.
Yeah, the same dysfunction, but because neurons are so highly dependent on energy, a very small deviation from normal supply of this energy can lead to very severe effects. And I'll show some very interesting data that we found, and I won't go to all the biochemical and molecular findings that we have, but only some anecdotal findings. Okay.
So you then now went into an experimental model, which is in rats, to see if you could simulate that and generate, if you like, an animal picture of what's going on in the brains of people with schizophrenia.
I would like to start with our human studies, okay? Because I think that this is to first show that indeed in human neurons there is a dysfunction of mitochondria, which lead to distorted neuronal development, distorted neuronal sprouting, and other things. If it's okay with
so, just to explain that for those who aren't afraid with neuroscience, what you're talking about here is that nerve cells in the brain branch out, and one nerve cell can connect to many thousands even of other nerve cells. They create networks, and it's how we think and remember and respond. And the richness of those networks is the richness of how our brains operate.
Yeah, we think that the connection between different regions, different neurons in different regions in the brain, this was what is the basis of memory, emotion, cognitive function, and even movements and movements. All our functions are affected by these connections through the brain. And if the neurons have poor spines or poor ability to connect, then or distorted ability to connect, we don't know if it's distorted more or less connection. It depends on the brain area. It can lead to abnormal behavior.
And you've got a dramatic shot, slide of what actually happens, so you can see quite dramatically in somebody's brain the difference between somebody with schizophrenia and a healthy person. We cannot look. I'm not looking at the brain of patients. I'm only looking at the development of neurons, because we think that schizophrenia may be a developmental disease. So we are following the development of neurons in dish in order to see what is the connection between the mitochondrial dysfunction and their ability to develop and to make mature of this neuron. This is what we do with neuronal cells, human neuronal cells. Imaging studies support our findings, because in imaging you have a window into the patient's brains, and you can see that there is abnormal connectivity. You can see that there is also abnormal energy at different parts of the brain. Because there are different kinds of imaging techniques that each of them is measuring different things. And what's the link between what you're finding? I mean, it's fine. You've shown you get an abnormality of the mitochondria. There's an energy problem, And what you're postulating is that affects the nerve cells and stops them branching and making the rich networks you need for good brain function. What's the link between that and human behavior and the sort of problems you see in somebody with schizophrenia, which is quite wide? I mean, probably people don't realize is that somebody with schizophrenia loses social skills, they lose a lot of basic abilities, they've got to relearn life when they start going into recovery, as well as the delusions that they get. It's quite a widespread problem that people have, very biological. But what's the link between what you've found and that sort of behavior?
Okay. In order to show a direct link between the mitochondria dysfunction and all this distorted neuronal function and behavior, what we did, we took healthy mitochondria and we injected them into a specific area, the prefrontal cortex, medial prefrontal cortex in the rat brain. And we did it in a rat model of schizophrenia, which is a maternal immune activation. We only injected once, a single transplant. We did a single transplantation, and this we did at adolescence, at an adolescent age, which is a very very specific time window in which the cortex maturation is finished. And we looked at our rats about 120 days later after this single transplantation, and we measured in them many biological functions, molecular functions, also cellular function, looking at the ability of neurons to sprout and to branch, and also we looked at the behavior of the rats. And what for us was a very astonishing finding, it was that this transplantation, single transplantation, after 120 days at adulthood, affected behavior related to behavioral deficits related to schizophrenia, such as attention deficits and movements disorders, and and not all the behavior, of course. And we have to bear in mind that it's a model, it's not schizophrenia, it's only a way to mimic several symptoms of the disease.
I might go to Ian now. I mean, this is fascinating because, you're first of all, there's branching, networking. You can see there's a lot of deficits in people with schizophrenia. And also injecting maternal rats, mothers. I mean, there's the hot theory in schizophrenia is that mothers might get infected with a viral infection and something goes on in the immune system of the brain.
So there's a number of aspects of Dorit's work which are worth emphasising. One is starting with humans. I really like to start with humans. There's a whole lot of neuroscience worldwide that starts with molecules or cells or circuits or rats and has no chance of ever arriving at human behaviour, like building the brain from the cell up. The good part is about starting with humans and going backwards, as Dorit's doing. You're picking something that's abnormal. So whether this abnormal in mitochondria is cause or effect, there's a problem with the energy translation system. The second really important bit is to attach that to development. So as you've just alluded to, Norman, nearly every developmental theory of schizophrenia says something goes wrong in the process of development. And one of the most commonly explored reasons is exposure of the child while in utero, while in the mother's tummy as a fetus, to immune activation, commonly by viral infection. Fascinating as we're now living in the COVID world, we have a huge amount of actual viral exposure going on. But looking back at past viral epidemics and exposures in countries, there's clearly some relationships with immune activation in certain phases of intrauterine development when the brain's developing. And then the next bit that Dorit's emphasised is you see the effect of that most then in adolescence. So the effect of that isn't immediately obvious in childhood, but in behavioural development when the immune system and the brain are undergoing another stage of development in adolescence. So a lot of Dorit's work fits with ideas about possible causes, the role of immune activation, the timing of that, and the critical aspect of this neuronal branching. And most theories of brain development and their relationships with behaviour these days talk about brain circuitry. How does that branching form into the circuits that control emotion, cognition, our physiological function? I think what surprises people in the mental health area continuously is actually that something is wrong at the physiological level, at the brain level, and at these circuitry levels, and certainly the developmental approach, and then the whole issue of branching and forming of connections, and we've all got to maintain them. We don't just get them, we've got to maintain them throughout our life to stay well.
So this is one of the theories why most mental illness comes on in adolescence, is that you've got an overgrown brain when you go into adolescence, and then adolescence and growing up is a process of pruning, like pruning a rose bush.
So Dorit made a key observation, which I think people don't realise. They think they use the most energy in the day running upstairs or doing anything else. Your brain soaks up a huge amount of total energy. In fact, you're growing as a child, it's doing so much connecting. If it kept going that way, you wouldn't have any energy to walk upstairs or do anything. And in fact, it becomes more efficient then in adolescence by pruning, by actually putting the right sort of synaptic connections, the right circuitry in place to be the most energy efficient, but also the most cognitively efficient. You don't want to have to think about how to speak. You don't want to have to think about how to walk. We do a whole lot of things automatically and energy efficiently. And that happens, that process happens as a long one throughout adolescence and early adult life to arrive at the most efficient and optimal cognitive state. And the brain's rest of its control over the rest of the physiology. It's driving your hormonal systems. It's driving your immune system. So in adolescence, the further development of the immune system and, in fact, of the brain systems together are critical. And we think they are some of the reasons why it goes wrong, why pruning goes wrong. Immune cells are running around the brain like electricians, actually driving wiring changes. So the microglia and other cells in the brain are very active. So not just the neurons themselves, but the immune cells are actually contributing to that pruning. So if the immune activation is off, you can be changing those circuits.
So, Dorit, are you finding any evidence of immune effects here?
I have a problem with, I know that what we did is inducing an immune activation in the mother during pregnancy, but the immune system, we don't see a very severe immune changes during in the offspring. Actually, it was also published by others that in this offspring there is no a very wide range change in the immune system. We do see that after injection of our mitochondria, healthy mitochondria, we do see a certain very mild increase in cytokines, which are markers of immune activation. We don't see changes, very profound changes in microglia activation, but still, and it's only in the short term, several hours or days after transplanting the mitochondria, but still, it may be that there is something going on during pregnancy. And what I said before is that maybe the cause has disappeared. The activation of the immune system and its effects on the immune system per se has disappeared. But what we see in the offspring model or in the schizophrenia patient is the effects or the long-term effects of this activation that has already disappeared. Like we say in the coronavirus, that we have very long.
Well, that's exactly the point I was going to make with Ian, is that you're almost describing long COVID, in a schizophrenia sense.
So there are interesting kind of things about many of the hypotheses of schizophrenia have a dual hit hypothesis, that there probably is a maternal factor. Something happens to alter the original structure, and then as Dorit said, it's then gone. But something else happens later on, typically activation during adolescence, and what that might be. So other theories have been other infections in childhood or in adolescence, like right now, the coronavirus. Or there is some other immune activation, some other antigenic exposure actually happens, and you get an activation. Interestingly, many of these maternal activation models are blocked by giving drugs like clozapine actually to adolescent rats.
So actually being an anti-schizophrenic drug.
Well, it's an antipsychotic drug, but it also has really strong immune modulatory properties and quite different to other antipsychotic drugs which just block dopamine. So it's quite different and the most effective antipsychotic we know. So there may well be some processes in the fetus, some other things that happen in adolescence that actually are the second hit. Now, a lot of debates. They may be immune activation, they may be drug exposure, they may just be developmental things that happen as a consequence of the prior insult.
So my next question to you, Dorit, is about reversal, because and just taking another mitochondrial problem, which is sarcopenia of aging. So in other words, as you age, you lose your muscles. And when you've looked at that, that's a mitochondrial problem. You've got old mitochondria in your muscles waste away almost in the way you're describing the nerve cells to be wasting away in terms of the networking. But what research at McMaster's in Canada has shown is that if you actually do hard exercise, even as an elderly person, you can rejuvenate your mitochondria. You actually end up with young mitochondria in your muscle cells. What traction have you been able to get in your research about reversibility?
Okay, what we think that in the mitochondria there is a core biochemical change in schizophrenia, in one of the very important complex that deliver electrons in downstream to other complexes in order to produce the energy. Okay, so there is a specific problem, and so the mitochondria are defected. So having more of the same will not overcome the problem. And actually, what we did, we took mitochondria from schizophrenia and put it into control cells and also into the brains of the rat, and we got worsening of the behavior, worsening of the sprouting of neurons in the culture. So the problem is that there is an inert deficiency or defect, and which we think we know which it is in the mitochondria of schizophrenia, and it's also it has also been found in Parkinson's disease, the same problem in the same complex that is very important for the energy production in the mitochondria. So this is maybe different from old people, because they have still very good mitochondria, very healthy mitochondria. So what they are doing with exercise, they are inducing the biogenesis of these healthy mitochondria. But in the schizophrenia, we know that there is a defect all over. We looked at different cell types. We looked at peripheral cells. We looked at neurons. We looked at other cells, astrocytes, and there is a defect in the mitochondria in the same complex. The effect of this defect is different because it depends on how, because the defect is very small, it's about 30%. So it depends how much this certain cell needs energy. If it's like lymphocytes, for example, they don't need so much energy. So a 30% decline in energy production does not do anything to their function. But if it's in neurons, it does.
And have you found any drugs that can interfere with this and make it
all the antipsychotics interact with the mitochondria. All of them. all of them. We have also the antidepressants have an effect on the mitochondria. So as I told you before, what we have done very recently, we looked at the possibility to use the different mitochondrial function as a tool to predict which drug will be optimal for a given patient to do a personalized medicine.
So on the flip side of that, there are certain drugs which are known to increase the risk of schizophrenia, either from scratch or in somebody who's susceptible. So cannabis, maybe MDMA, amphetamines such as ice. Have you tried to induce the abnormality using some of those drugs?
No, we didn't do it. We didn't look at the effect of these drugs on the mitochondria, but we know that most of them are increasing dopamine, and we have shown that dopamine itself can also regulate mitochondrial activity. This is also our very first studies that have shown that dopamine itself can regulate mitochondrial activity. So all these drugs also have, I don't know if they have a direct effect on the mitochondria, but certainly they have an indirect effect by increasing dopamine.
One of the things that Dorit's alluding to in terms of working out which drugs work in mental health is nearly all the drugs that have a really positive effect are associated with increased neurogenesis or generation, particularly of new synapses, and to some degree, potentially generation of new cells, particularly within the hippocampus. So, this is an interesting kind of idea, and they are certainly energy-dependent processes that actually...
Yeah, but the drugs are terrible. I mean, they don't...
Now, we're going to have a debate about that. When you say terrible...
Well, they control the psychosis and the delusion, but they don't necessarily help with recovery.
I'm going to argue with you about that for exactly this reason. I think that's actually wrong. That actually, if you look at the longer-term effects, if a drug
What
does
do you think
not...
you are, a professor of psychiatry or something? Really?
Well, I think what people confuse is two things. And go back to Dorit's point, why neuroimaging has been so interesting. And we've seen more studies of this recently. In fact, those people who are on medications longer term have evidence of greater synaptic connection and actually preservation of brain cells compared with those who've got those disorders untreated. And I think the thing confuses two things. One is symptomatic relief. Okay, when people are very unwell with acute psychosis, dopamine blockade makes a big difference. When people are very unwell with acute depression, relief of those symptoms may occur early. And that seems to be more biochemical in its nature. And that's what you're saying is only symptomatic. But actually, longer term, if you go to what is really effective longer term, if those drugs are not associated with a neuroplasticity, neurogenesis effect, they appear to be not very good treatments. And I think this has become the more interesting kind of area. Which drugs, whether we call them antipsychotics or we call them antidepressants, you see similar things actually with anticonvulsants. And those which are associated with neurogenesis, actually, I'm going to argue with you, I actually think they do restore the brain longer term. And it's really interesting. It's not the same time course as symptomatic relief. So all of our drug regulatory things look for symptomatic relief. In a sense, that's like taking an anti-inflammatory. It's like
you stop the delusions.
You stop the depression, you stop the sleep disturbance, you stop the delusions, you stop the abnormal behaviour. That's symptomatic relief. And that's how we license drugs for efficacy. But there's this other issue, really, what's happening to the brain. And what we've lacked, what Dorit's alluding to, we've lacked good brain markers of what's happening to the brain. If you think what's happening in multiple sclerosis, and I'm pointing across the corridor here where we treat MS, we've got good white matter changes, immune changes. You can see what's happening to the brain in relation to the immunotherapy, not just the symptoms, and therefore the course of illness. And that's where we've really struggled in psychiatry, is to have good correlates of brain recovery to match actually person recovery, not just short-term symptomatic relief.
The neurogenesis is mostly related to antidepressants and less so to antipsychotics. And I quite agree that, although I'm not a clinician, I quite agree that the severe side effects and that about 30% of the patients do not respond to the drug, we cannot we cannot lay on, on our backs and say we have good drugs. We still need to find better drugs for the patients, and this is my goal and wish, that I'll find a new target that may be a better way for treating.
But also, I think we've got to be careful. We've got to say, well, but we have much better drugs now than we had 40 years ago. And before, 50 years ago, we had no drugs at all that really were at any specific effect. The first discovery of actually effective antipsychotics, which were based around dopamine blockade, transformed the lives of people who were otherwise had enduring psychosis. The first discovery, subsequent to that, of antidepressants that really worked from Ipamid onwards. But all of those had a lot of side effects. And for those of us who are older psychiatrists, what we first prescribed were terrible in terms of their side effect profiles, in terms of inducing things like Parkinsonism and terrible motor side effects and terrible other effects from the antidepressants. Now they're better. They sure still have a lot of side effects, a lot of them longer term now, sort of different. So what we're struggling with, but I think it's actually probably the combination of medications we're going to need. So in a lot of the work I'm associated with early intervention, we're trying to treat the early symptoms to turn the illness off. But we're really interested in the long-term brain outcome, the neuroprotective effect, or what will be required. And there's a lot of interest in immune modulation, neurogenesis, other pathways, which may not provide great symptomatic relief, but may be important. So I think in the future, we're going to be asking different questions. What combination of treatments turns the symptoms off with the least possible side effects? But in the longer term, which ones are best for the brain? Because there's ongoing processes. Whatever started this process in many people with chronic depression or with psychosis, it continues. And that we've got a longer term brain process we need to turn off and hopefully restore to better circuitry for better behaviour.
So Ian, Dorit, thank you very much. That's fascinating and a lot and full of promise. So thank you all. Thank you both very much. Fascinating discussion.
Hi, welcome Dorit and Ian again. In case you were wondering, or I guess this was a pre-recorded session that Ian and Dorit kindly attended a few days ago, and therefore I apologize if some of the subtitles weren't 100% aligned. This is a service that YouTube provides, and I promise you there is no such disease called parking disease. This is something we were trying to do to enable the webinar to be more accessible to more crowds. But we have them, Ian and Dorit, live with us now. So thank you for joining us. And this is the opportunity for you, our audience, to ask your questions and pick the brains of the experts that are with us now. We have quite a few already, and we had a few that were lined up before the event that were submitted prior. So with no delays, if you're okay with it, Ian and Dorit, I might start with the first question, which might be to Ian, which was submitted a bit earlier today. So given the brain's high dependency on energy, how does this actually manifest in schizophrenia?
I think Dorit's raised two issues. Dorit's looking at the fundamental energy mechanisms through mitochondria in which they may be disturbed. And that would then affect a whole range of cellular functions on an ongoing basis. I think what we were talking about earlier on is how those energy components play out in terms of formation of nerve circuitry and cells. So I think there's a whole lot of different levels at which changes to cellular function affect the way cells are then able to grow and form circuits that we were talking about a lot through those particular issues. So this is potentially relevant to specific diseases like schizophrenia, but it may be relevant to a wider range of brain-related conditions, certainly psychiatric-related conditions, because there probably are a range of different factors that can alter the way nerve cells generate, form new connections, and maintain the critical circuits that we all rely on to be able to think, to breathe, to emote, to remember, all of those brain functions we take for granted.
Excellent. Dorit, I've got a question. I'm going to refer it to you. It's from Sean Murray, who is from the MITO Foundation, which is the Australian Mitochondrial Disease Foundation. And he's asking, is schizophrenia is a primary mitochondrial disease, or is that so in some cases?
I can't say that it is a primary mitochondrial disease because it's a very complex disease, and we don't know actually which factor is prior to the other. And so I don't know if it's a primary mitochondrial disease, but I surely know that there is a certain defect that is in the mitochondria, in the first complex, and the whole function of mitochondria is dependent on the surrounding cells and the surrounding environment, and thereby it may, as Ian said before, lead to different disorders, because it's not specific only for schizophrenia, but there is a specific characteristics of dysfunction in mitochondria that are related to schizophrenia. For example, in Parkinson's, the whole situation is a little bit different, although there is the same complex which is defected, but there is also there is the surrounding of the cells, and for example, increase the ROS activation, reactive oxygen species there, that push the whole scenario to a different outcome.
Right. All right. Thank you very much. The next question is, I will let you choose who's going to answer that because I'm not quite sure. But what role do hormones play in the adolescent stage, which may affect the mitochondria? Ian, do you
I'm
want
going to leave that one to Dorian. That's for Dorian.
I try to answer it. It's a very good question because we actually don't know exactly. We know that mitochondria is a hub of a cell, so it's involved in all these processes, but I cannot tell you how exactly and whether there is a connection between the hormones and the mitochondria. They surely affect the mitochondria, but how it is related to the adolescent state, the changes during adolescence and mitochondria, I don't have any good answer for that.
All right, thank you for that, Dorit. The next question is, what role do hormones play? Oh, no, I'm sorry, I just asked that one. Is it possible to anticipate and antidepressants also have indirect effect on mitochondria instead of direct?
We have shown that they have a direct effect on mitochondria, but they may also have an indirect, because we have to look at the whole cell, or the whole, not only the whole organ, but the whole cell as a network. And if something gets wrong in this network, the whole equilibrium is changed, and that may lead to different outcomes. It may lead to depression. It may lead to diabetes, for example, if it's in another organ, or it may lead to schizophrenia. So it is very, very difficult to take out each factor and look at it separately and not in the whole context. I think this is my best answer to this question. So we show that there is a direct effect of the antidepressant on the mitochondria, but they may have a lot of other effects. For example, we know that SSRIs are interacting with VDAC, which is the main ion channel in the mitochondria. We know that they are interacting with other complexes there, but still they have other effects that can change the whole signaling pathway in the cell, which can also affect the mitochondria, for example, calcium concentrations and so on. So they may, it's more than one factor that is involved in this complex outcome.
Great, thank you. Ian, I'm gonna address the next one for you. Does anorexia, therefore the abnormal deprivation of energy, create a risk for schizophrenia or other mental illnesses beyond the illness of anorexia?
I'm not aware that there's any evidence that anorexia itself is a precondition for psychotic disorders. Unfortunately, anorexia is associated over time with a lot of other mood disorders and other difficulties, and very sadly with premature death in proportion of cases. So anorexia is a severe enough illness on its own without creating an additional liability to psychosis per se. Quite often, what you do see when people with anorexia actually are re-fed, get back to normal body weights, that then actually other depressive disorders and other mood disorders become much more obvious at normal body weights when people recover. So often there's quite a complicated set of issues between their eating state and emotional regulation, but I'm not aware of any specific link with risk to psychosis.
Great. Any of you want to take this one? What is the long-term effect of the brain with taking new and I guess existing antidepressants?
Yeah, I wouldn't mind taking that one. I was very, very glad to have that argument with Norman about, what are good drugs and bad drugs. In the modern world, partly because in the modern world, what gets called a bad drug gets called a bad drug fairly quickly. And I think, for those of us who are old enough, sort of an idea about what it was like to have very few treatments, and then the treatments we had a lot of side effects, a lot of really significant side effects. People raised issues about Parkinson's disease in the question and answers, other severe longer-term motor effects of the older drugs. The newer antidepressants, which have been in existence really since the 1990s, and even the antidepressants that preceded those from the 1960s, the antidepressants per se are not associated with long-term adverse brain changes. Now, the antipsychotics, there's been much more argument about their relative benefit or not longer term, and whether there may be problems associated with antipsychotics longer term, and certainly other brain changes and the development of Parkinsonian-like syndromes. So with the antidepressants, no. I think what I was alluding to is the interesting thing is the reverse, whether in fact they have these neurogenesis or promote brain regeneration, brain reconnection, where that has been diminished by the illness itself. There's a really interesting question in the Q&A from Hickie about, will we be able to make some sort of compromise here? Could we promote this neurogenesis or brain recovery effect without necessarily getting the side effects from the current drugs? The point I was trying to make with Norman is it's not just the neurogenesis. You still have to get symptomatic release. Let's not forget how bad it is to have these illnesses in the first place. If you have severe depression, if you have bipolar disorder, if you have psychosis, it's not exactly as if you can carry on with your life in a normal way. So the symptomatic component, which in the short term appears to respond to biochemical type interventions, not dependent on growing new nerve cell connections, isn't the same. So these are the kind of really interesting issues about where pharmacotherapy or medical therapies will go in the future. What combinations of medical therapies might give both the best short-term symptom relief with the minimum side effects, while also promoting, or through different mechanisms, promoting longer-term brain recovery. Of course, there are other non-medical things, physical activity, exercise being one, for example, that have been shown to increase, brain recovery in these particular areas or neurogenesis. Socialization, being in contact, being engaged in activities and intellectual activities like we are now. So there are other things that drive brain synaptic connections in addition to medicines. And I think that's one of the other important kind of areas in this area. And there are other dietary and metabolic factors that may well be relevant. So I think these remain very interesting areas of brain research, trying to work out the optimal combination to treat illnesses, but to end up protecting brains or having brains recover best that they might from whatever insult has led to the onset of the illness in the first place.
Excellent. Thank you, Ian. I've got another one for you because I know you deal a lot with youth mental disorders, and we've had a question that came in prior to the session. You mentioned that poor memory in schizophrenia patients, so poor memory is sort of very typical in schizophrenia patients. However, there are exceptionals with people with phenomenal memory. If you can share some insight on that.
Yeah, probably worth clarifying. I was talking about disturbed cognitive function, a whole range of cognitive functions in the acute phase of illness. So schizophrenia is not an illness per se where memory deficits is a primary problem, like it is in dementia, like in Alzheimer's disease, where memory is one of the core kind of features. So the problem with memory in illnesses like schizophrenia in the shorter term is a problem with attention and with focus and the way we lay down memories rather than the memory apparatus being disturbed. Again, restoration of cognitive function very much depends on treating that illness in the first place. So people who have high cognitive function beforehand and have particular cognitive attributes, good memories, good other factors, high intelligence, if you treat the illness successfully, may well return to having the same level of cognitive function that they had prior to the illness.
Okay,
but I might say, just on the early intervention, that depends actually on treating the illness early and effectively. So if you go back, 100 years to when the schizophrenia was originally described, the way we now understand it, it was seen as a dementia precox, as a onset of a dementia-like illness in young people, because people just ran a deteriorating course. Things just got worse and worse when we didn't have effective treatments.
Excellent. Next question is from Sarah Cummings. Do you think that this new knowledge will in the future lead to a way to screen children for the potential for them to develop schizophrenia in adolescence, or at least the opportunity for it to happen?
Propensity. I see the propensity word.
Sorry.
I don't know. I'm not sure from the mitochondrial point of view, Dora. Do you want to comment from the mitochondrial point of view before I take off into the genetics separately?
Okay. I think that maybe because the mitochondria are so flexible and dependent on the environment, I don't know how we would be able exactly to know, but certainly it may be, because maybe also, as you said, there are certain together with genetics, together with things that are more stable and not so time dependent. The changes are not so time dependent. Maybe together with this information, the functional changes, which are more, more important for the state of the person, may together lead us to a biomarker or an endophenotype that will help us to predict whether somebody will have schizophrenia or not.
So just on the genetics at the moment is really interesting, because after about 30 years of research, people are starting to generate what are called polygenic risk scores for all of the major psychiatric illnesses. So when you look across the population, you see combinations, and these are combinations of hundreds of genes, not simple 1 or 2 genes, that increase the risk that you might be at risk of schizophrenia or bipolar disorder and now depression. That's not going to lead to screening tests per se, because it will only tell you about the vulnerability, the possibility, not that you inevitably will get that illness. But where it may become interesting is if you have come from a family where you have some known past history across generations in your own family in particular, who might be at risk and who might be at more risk. And whether in some of those situations, much as we see in breast cancer, we see in other areas where families are at high risk, that individuals within those families want to know, how much more risk am I at? And are there things I can do? I think, and this is a matter of very active research at the moment, could you combine that with an earlier intervention approach? So that if teenagers were starting to develop symptoms and they also had a high genetic risk score, would you actually intervene earlier? Would you consider certain kinds of treatments at an earlier stage? So this is a very active area of research at the moment. It's too early, and I don't think we'll ever be moving to a situation of population level screening, but we may well be in situations where people have other reasons to believe that they are necessarily at high risk and want to take more active strategies earlier.
Great. Are there any other nutritional contributors to poor neurogenesis, like vitamins or mineral deficiencies?
Starvation is not good for you. So where we've had issues of starvation and people had major vitamin deficiencies, and things that we used to have, for example, iodine deficiency, and we see lack of thyroid hormone development in other areas. There are certain kinds of deficiencies that, yes, do affect brain development and cognitive function, but they're not in relation to normal diets and normal development. Obvious that there are other dietary explanations. Again, a lot of interest worldwide in what food things might be particularly good. I mean, generally speaking, vegetarian type diets and everything else be good for your brain, along with many other parts of your body. And there's a lot of interest in whether there are particular gut-related factors that might also be influencing brain function, you know, as a consequence of what we eat and the lifestyles that we have, but not in terms of very specific associations. Where there have been major deficiencies, either through starvation or through other individual differences, at times they may be relevant, but not a common cause.
All right, there's a COVID question here. You didn't expect to get away without it.
Yeah.
Do we anticipate an influx in schizophrenia or psychosis in about 10, 15 years following the current COVID crisis in adolescents at that time who are currently or recently in utero or exposed to COVID?
I don't know. What do you think, Dorit?
You know what it has been said about people who try to be prophets, and they say what will happen, because nobody knows about this virus enough in order to say whether it will lead to higher cases of psychosis. But you know, if it's like other autoimmune disease like the influenza or something like this, it may. We actually don't know, but we hope that we'll have some good cures in order to prevent this.
It certainly is. Certainly is a concern. Anytime you have a major set of febrile illnesses, we were discussing earlier on the cytokine release, a lot of immune products being released. Certainly, if they are exposed at certain times of brain development in utero, it's an issue of concern. But different viral epidemics have and have not been associated later on with spikes in the cases for psychosis and other disorders. So I'm afraid to say it's one of those things we will just have to wait and see without clearly knowing whether this particular pandemic, certainly the severity of the illness associated with the sort of febrile illness that we've seen and the extent to which people are being left with sort of long COVID syndrome, certainly suggests that the immune activation has been very high. And just as with all other pregnancy-related situations, it'd be one of concern, but the real answer, we don't know.
We'll book you in for 10 years from now. So
15, don't rush.
Yeah.
All right. Does treating depression early is an effective way, as many try to avoid it, giving drugs until a situation is unavoidable?
Yeah, so the depression situation in some ways is a bit easier than the psychosis situation. When people are acutely psychotic, it's very hard to avoid treatments with medicines. If people's whole cognitive function, their whole behavior and emotion component becomes so disturbed that actually there's often little choice but to use the medicines that we have to relieve that state. With depression, and depression in younger people, generally speaking, we would try and treat it in the first place with psychological therapies of various sorts first. And alongside other kind of issues around sleep-wake cycles and around physical activity and around social engagement. So, you know, if possible, and certainly in the majority of situations, medicines would not be the first choice for most depressive illnesses in teenagers. However, if it is more severe and it is ongoing, then yes, and a lot of our work is in that kind of area. And I think this is kind of these trade-off kind of issues. Which is worse? In my view, it is certainly worse to have the untreated illness. If you look at the brain sort of work that we're doing, the brain imaging work that we're doing, untreated illness is worse than treated illness. If you do respond to psychological therapies and other lifestyle interventions, great. For those who don't, who have more severe illnesses, or those who go on to have recurrent or persistent illnesses, then I think the news is actually considerably better than is often promoted. Is actually, from a brain point of view, it's often better in terms of actual people's return to school and to work and leading more productive lives. It's better. I think with younger people, in particular, what we're trying to do is treat the episode of illness, not necessarily have to use medicines for a prolonged period if they're not necessary.
All right. Can long-term psychological stress factors cause an increase in mitochondria activity, and thus leading to a possible cause in schizophrenia? And what is the damage long-term if exposed to psychological stress factors? Dorit?
Okay. Okay, I think Ian will add to it from the clinical point of view, but what we know is that long-term stress can change the mitochondrial activity. We did a very nice experiment in which we harmed one of the factors that affects mitochondria, and we did a very short and a very long-term stress period. We exposed the rats. It all was done in animals, not in humans, of course, and we exposed them to short and to long-term stress. And what we wanted to have a model for schizophrenia, but what we got actually was that the short-term stress exerted depression-like symptoms in the rats, and the long-term stress exerted mania-like expressions or features, both behaviorally and biochemically. So, and we know that, and also the mitochondria were altered in both short and long-term stress. So what I can say is that I think that when I looked it up in the literature, long-term stress effect is much more associated with depression and mood disorders, and less so with psychotic disorders. But still, it may affect some features of the schizophrenia disorder, as we know that it's not, there are very different types of clinical manifestation of the schizophrenia. So this is what I can say about it, and that for sure, long-term stress or even short-term stress affect the mitochondrial function. The question is how the mitochondria can regenerate after such stressors.
All right,
Certainly,
thank you. Oh, Ian, go ahead if you want to add something.
I think it's good that people have raised the long-term stress, because I pick up Dorot's point. I mean, we all run into acute sorts of stresses variously in our lives, most of which, unfortunately, not life-threatening or overwhelming. They're not so much associated with mental health problems, but chronic stress is. So in humans and in most animal models, chronic stress is really the problem. And then the biochemistry of that, how it is that your stress response through your hypothalamus and pituitary hormone systems drive your cortisol response, how they release stress hormones. And if that happens, which is meant to only happen short term, when that happens long term, it's bad. It's bad for your brain, it's bad for your immune system, it's bad for all of these. If you can't turn stress off, there are a lot of long-term biological consequences on the brain, on the hormonal system, on immune systems, all of which are bad for your health. So, the thing for humans is when they run into difficulties, how quickly can we actually turn those problems off and reconstitute, having been faced by the threats and stresses that we particularly have. So they are certainly the main models that we have for the onset of the major mood disorders, things like depression, long-term anxiety, post-traumatic stress. I see mentioned a lot in the Q&A. These are the sort of models, and then they are having very negative effects, very negative feedback effects from being permanently turned on when they really should be only turned on for a short period and then turned off. So, and you create a very dysfunctional system. And the feedback then on the brain, in terms of neurogenesis, in terms of synaptic connection, is all bad. It's all negative. It's actually, you need to turn it off. You need to, it's interesting, as does not sleeping and a range of other things. A whole lot of things you need to do, which we turn on in order to respond to the environment in appropriate ways, that we also need to turn off to regenerate, to be able to carry on in the future.
Excellent. So we're coming towards the end of our hour. I'm going to ask you a last question that came from the audience, is whether you find any significantly different research result and outcomes in people with pre-existing organic conditions or deficiencies. Ian, do you want to take that one? Or
I just missed it. Could you just repeat the question?
If you find any significantly different research results and outcomes in people with pre-existing conditions, organic conditions.
I'm not entirely sure what the question's driving at. one of the things that does occur is that mental health problems often occur in the context of other physical health problems and other difficulties. And when they do, they're more complicated. So people with other physical health problems, certainly those affecting the brain, are more likely then to develop other mental health problems. But it's also true the other way around. Young people with mental health problems go on to develop many, many more physical health problems, early cardiovascular disease, other difficulties, and like, for example, people with depression are at increased risk of developing dementia later in life. So they're actually at increased risk of developing other brain diseases. So probably what you're seeing is a bunch of processes that are shared across what we classically call mental health and physical health, affecting the brain and affecting the whole body. Not good either way. The earlier you get any of these problems and you treat them, the better your health is likely to be in the longer run.
Yes, and there has been a very rich literature about the connection between cardiovascular diseases and depression, Parkinson's disease and depression. So mental disorders are not standing alone. This is one of our main beliefs, that the mental disorders are exactly as other general disorders, and they are connected. They are truly connected. Also, fever with depression and a lot of other physical diseases that are well connected with mental disorders. It has been seen that sometimes the mental disorder precedes physical disease, and sometimes it's the opposite.
All right, excellent. Thank you very, very much. We've reached the end of our hour, unfortunately. There's still a lot of questions out there, and with your permission, I might still ask you later and see if we can come back to our audience in a different format at some point. I wanted to firstly thank our moderator today, Dr. Norman Swann, Professor Dorit Ben-Shachar from the Technion in Israel, and Professor Ian Hickie from the Brain and Mind Center, part of Sydney University. Thank you very much to you. It was very, very interesting. I wanted to also acknowledge our partners today again, Meeting for Minds, which is a not-for-profit with a core mission to enhance any scientific research associated with mental illness. Importantly, Meeting for Minds advocates for cooperative inquiry programs where people with lived experiences of mental illness become involved with specific brain research projects as partners as opposed to subjects. Of course, to thank Sydney University, and in particular the Brain and Mind Center, which Professor Hickie runs, and Professor Hickie's research into youth mental health and technology. And lastly, the Technion, and in particular the Research Laboratory of Psychobiology, the Department of Psychiatry, the Ruth and Bruce Rappaport Faculty of Medicine at Rambam Healthcare Campus at the Technion, Israel Institute of Technology. Without the support of our community, the research that Ian, Dorit, and their colleagues conduct could not be possible. So we encourage you to consider supporting them. And we'll have a little banner at the end where you can, who you can contact if you want to hear more or find out how you can support or just learn more about their research. Next month, we'll be hosting a very exciting webinar on cyber threats and the dark web with a Technion alumni who is now the CEO of one of Israel's fastest growing cyber tech companies, along with the head of cyber at Canva, which obviously the Australian viewers here know is an incredible success story, a tech company here in Australia. We'll send you further details on that shortly. And lastly, please join me in wishing the Technion a happy anniversary of the laying of the cornerstone, which was 109 years today. And of course, for Israel's independence, 73rd independence, which is coming up this Thursday. Lastly, when you sign off, you're going to have a very quick survey, which take you no longer than a minute to complete, which will really help us to improve our webinars in the future. So if you can take up the time for that, we'll really appreciate that. Until next time, thank you again. Take care.
Thank you. Thank you very much.
Thank you, Ian. Thank you, Dorit.
Goodbye.